Hello everyone, welcome to week 9 TA session for drug delivery principles and engineering. So we'll cover some assignment problems based on the topics covered in the previous week, and the topics are immune and blood response to materials, which includes immune systems, the complement system and blood clotting and hemocompatibility of materials, adaptive immune response and vaccines. Based on these topics, we'll discuss some previous year questions now. So here is our first question. Which of the following options represents the correct chronology of events in inflammation? Options are vasodilation, edema, influx of leukocytes, clearance, healing. Second option, vasodilation, influx of leukocytes, edema, clearance, healing, vasodilation. Third option, vasodilation, edema, clearance, influx of leukocytes and healing. Fourth option, an influx of leukocytes, vasodilation, edema, clearance, healing. Fourth option, vasodilation, influx of leukocytes, clearance, healing and edema. Next option, an influx of leukocytes, edema, vasodilation, followed by clearance and healing. Or vasodilation, edema, influx of leukocytes, healing and clearance. Or last option, an influx of leukocytes, vasodilation, edema, clearance and healing. So which of these would correctly represent the chronology of events in inflammation? So the correct answer here would be vasodilation followed by edema, followed by influx of leukocytes, then clearance and then healing So vasodilation, so following a tissue injury, the local blood vessels rapidly dilate due to inflammatory mediators like histamine, prostaglandins, nitric oxide, and it increases the diameter of the capillaries, leads to redness and increasing the blood flow to the site. This causes redness and heat and early cardinal signs of inflammation. Next is edema. This increase in capillary permeability leads to influx of protein-rich fluid and cells or exudates called as edema. And these proteins leak into the interstitial tissues leading to swelling, and it would contain this protein-rich fluid, albumin, immunoglobulins, complement factors and other proteins which are critical for defense and healing, and would form a swelling near the site of injury called as edema. Next would be the influx of leukocytes. So neutrophils, influx of leukocytes and phagocytic cells into the tissues. So neutrophils and other leukocytes are recruited from the circulation and they migrate into the inflammatory tissue through chemotactic signals. They enter via margination, rolling, adhesion and transmigration, and these cells phagocytize phagocytose the pathogens and clear debris from the site, which is what forms the next step, that is the clearance. So the leukocytes then engulfs and destroy the invaders, bacteria debris through phagocytosis, producing reactive oxygen species and lysosomal enzymes. And this phase is characterized by pathogen elimination and tissue debridement. Following this comes healing and tissue regeneration. So once the inciting stimulus is neutralized by clearance, the inflammation terminates through active resolution pathways. Lipoxins resolve in apoptosis of neutrophils, etc. And macrophages clear dead cells, and the tissue repair begins through fibroblast proliferation, angiogenesis, and remodeling, basically formation of new capillaries and the scar tissue. So this will form the entire chronology of events, which starts from vasodilation, followed by edema, followed by influx of leukocytes, and then clearance or healing. So this would be the correct answer. Moving on to the next question. So the next question is, the three arms of the complement cascade converged at a protein complex known as, options are C5 convertase, C3 convertase, C5a, C3a, C3b, C1q, C8, C9. Which of these converge at protein complex? Which of the three complement systems converge at which of these protein complexes? So the correct answer here would be C3 convertase. So there are three arms of the complement cascade systems: the classical pathway, the alternative pathway, and the lectin mediated pathway. And all of these, they basically, all of these converge at the formation of the protein complex known as the C3 convertase. The C3 convertase then cleaves the complement protein C3 into C3a and C3b, a critical amplification step that enables opsonization, inflammatory signaling, and ultimately leads to the downstream formation of membrane attack complex via a C5 convertase assembly. So these pathways basically differ on how they are activated, but all of them will converge at C3 protein complex, C3 convertase protein complex. So C3 convertase will be the correct answer. Moving on to the next question. Question is, which of the following approach can be employed to reduce complement activation on a biomaterial surface? You have to select two correct options here. And the options are reduce surface electrophiles, reduce surface nucleophiles, binding to C5b protein of the complement cascade, binding to C3a protein of the complement cascade, heparin adsorption to the surface, adsorption of C3 convertase agonists, coat surface with fibrin. So the correct answer here would be reducing surface nucleophiles and heparin adsorption to the surface. So there are three main ways to minimize the complement activation by materials. One is less surface nucleophiles or reducing the surface nucleophiles, which reduces the binding of C3b to the surface. So decreasing the nucleophilic functional groups, for example amine or hydroxyl groups on a surface, can reduce complement protein binding and subsequent cascade activation by limiting the sites for C3b covalent attachment. So reducing the surface nucleophiles will reduce the sites of attachment of C3b and subsequently inhibit further complement cascade activation. Next, binding to C5a, thereby reducing patient exposure. C5a is one of the primary causes of inflammation and other deleterious reactions. So it can be separately ablated or binding it on a surface such that its activity is reduced. By using these approaches also we can reduce the complement activation. From the question, the next option that was correct was heparin adsorption to the surface. So heparin coatings are a well-established method to reduce both complement and coagulation activation on biomaterials by inhibiting cascade components, and it acts through another protein factor H to inactivate C3 convertase. Moving on to the next question. Which of the following factors can cleave fibrin in vivo? Repeat the question. So next question is, which of the following factors can cleave fibrin in vivo? And the options are plasmogen, fibrin, fibrinogen, plasmin, prothrombin, thrombin, factor 8 and factor 12. So the correct answer here would be plasmin. The factor that can cleave fibrin in vivo is plasmin, which is option 4. And plasmin is the main enzymes that is responsible for fibrinolysis, actively breaking down fibrin clots within the body. This plasminogen is the inactive precursor, and while thrombin, prothrombin, and other coagulant factors are important for forming the fibrin from fibrinogen, they do not break down fibrin. Fibrin and fibrinogen themselves are substrates, not enzymes. So the correct answer here would be fibrin, which is the only factor that can cleave fibrin in vivo. So the correct answer would be plasmin. Moving on to the next question. You have to pick the incorrect statement here regarding cytokines. You have to select two correct options here, two incorrect options here. So from the following, which of these would be incorrect regarding cytokine? The options would be cytokines are small lipid molecules that are released by various cells in the body, usually in response to an activating stimulus. Second option, cytokines can act in an autocrine manner so that it affects the same cell type that releases the cytokines. Third option, cytokines can act in a paracrine manner so that the cytokines affect the behavior of adjacent cells. Fourth option, some cytokines can have enhanced bioactivity when conjugated to molecules like polyethylene glycol. Fifth option, cytokines are highly potent proteins that bind to the conjugate that They bind to their cognate receptors and induce downstream signaling cascades in the respective cells. Last option would be cytokines in their active forms are always pure peptide chains without sugar lipid moieties. Which of these are incorrect regarding cytokines? So the incorrect options here would be cytokines are small lipid molecules that are released by various cells in the body, usually in response to an active stimulus. And last option, that is cytokines in their active forms are always pure peptide chains without sugar lipid moieties. Cytokines are basically small proteins that are released by various cells in the body, usually in response to an activating stimulus, and induce responses through binding to specific receptors. They can act in an autocrine manner, affecting the behavior of the cell that releases cytokine, or in a paracrine manner, affecting the behavior of adjacent cells. Some cytokines can act in an endocrine manner, affecting the behavior of distant cells, although this depends on their ability to enter the circulation and on their half-life. So based on this, cytokines are small lipid molecules that are released by various cells in the body, usually in response to an activating stimulus, is false because these are not lipid molecules. There are small proteins or peptides, and in some cases glycoproteins, so they are not classified as lipids. Next is cytokines in their active forms are always pure peptide chains without sugar or lipid moieties. So as I said in my previous option, this is also incorrect because many cytokines can also be glycoproteins, that is not pure peptide chains but with some modifications with glycosylation, that is either can contain sugar moieties or some other modifications. So there are many cytokines, especially in the interleukin and interferon families, which have this sugar moieties, and basically they are glycosylated, and modifications which can influence their biological activity and half-life. So these two options will be the incorrect options among the statements for the cytokines. Moving on to the next question. Inflammation facilitates the onset of extravasation. So you have to select the correct option here. Two correct options. Options are: it refers to the process of macrophages clearing the dead cells in the vicinity of damaged tissue. It refers to the option 2. It refers to the process of phagocytosis of pathogens that would have entered the body through an open wound. Option 3. It is the upregulation of adhesion molecules on endothelial cells followed by the release of chemotactic factors. Option 4. Extravasation is the partitioning of cells into different blood vessels based on the concentration gradient of the chemoattractant. Option 5. During the process, the velocity of blood flow decreases and allows for cells to interact with endothelium. Option 6. It involves the process of migration of neutrophils through the endothelial layer. Which of these are correct options? So the correct options would be during the process, the velocity of blood flow decreases and allows for cells to interact with endothelium. And another option, it involves the process of migration of neutrophils through the endothelial layer. So extravasation, during the extravasation there is decreased blood flow which allows the leukocytes to interact with endothelium. And then rolling adhesion, which is very weak interaction initiated by interactions between carbohydrate moieties and selectins. It allows for strong interaction between the endothelial ICAM1 receptors and immune cell receptors LFA. And then rolling arrested and extravasation begins. So MMPs are secreted to facilitate transversion of the basement membrane. So the correct options during the extravasation processes would be that the velocity of the blood flow decreases, which allows for cells to interact with the endothelium, and it involves the process of migration of neutrophils through the endothelial layer. So let's move on to the next question. What is the role of platelets during blood clotting? You have to select two correct options here. Options are stimulate the clotting of blood and wound healing, formation of a clot by reacting with plasminogen, release variety of chemical mediators that promote clotting and healing of damaged tissue, transport of oxygen, generation of pro-inflammatory molecules, generation of pro-fibrosis molecules, or formation of giant macrophages. Among these, which two options are correct for role of platelets?
Natural.
So the two correct options here would be stimulate the clotting of blood and wound healing, and release variety of chemical mediators that promote clotting and healing of damaged tissue. So the role of platelets during clotting would be to react with the extracellular matrix proteins when blood vessels are severed or with biomaterial surfaces, to release a variety of chemical mediators from granules, or to stimulate coagulation and wound healing. So platelets adhere to the damaged blood vessel sites, aggregate and release chemical mediators to initiate and promote clot formation and tissue repair. And they secrete factors like uh, basically several chemical mediators that promote clotting and healing of damaged tissues, like ADP, thromboxane A2, serotonin, and growth factors that amplify the clotting and support healing. But platelets do not cleave plasminogen or transport oxygen or form giant macrophages, and their role is primarily in clot formation and promoting healing rather than generating pro-inflammatory and pro-fibrosis molecules. Moving on to the next question. We wish to generate an osteoconductive implant. We wish to generate an osteoconductive implant by coating it with fibronectin. For this purpose, an aqueous solution of fibronectin was prepared. And assuming the surface of the implant is positively charged, how should we change the pH of this solution so that is large amount of fibronectin is attached to surface? The pI of fibronectin is 5.75. So what would be the correct option here? Make pH less than 5.75, make pH more than 5.75 at 37 degrees, make pH more than 5.75 at 25 degrees storage temperature. Next option, do not change pH, or make pH 5.75 at 37 degrees, or make pH less than 5.75 at 37 degrees, or make pH less than 5.75 at 25 degrees storage temperature. Which of these would be the correct option here? So the correct option here would be make pH more than 5.75 at 37 degrees. So to maximize the fibronectin attachment to the positively charged surface, the fibronectin molecule should have a net negative charge. And to facilitate the electrostatic interaction between the fibronectin negative charge and the implant positive charge. And since the pI of fibronectin is 5.75, at pH values above this pI, fibronectin will have a net negative charge. So we have to make the pH more than 5.75 at 37 degrees to promote maximum attachment. Since the solution pH should be above the pI of the fibronectin to carry a negative charge and bind effectively to the positively charged surface. Moving on to the next question. Which of the following is not a physiological event occurring during inflammation? You have to select two correct options here. 1. Vasodilation, increase in diameter of the capillaries of local vasculature. 2. Increase in capillary permeability. 3. An influx of protein-rich fluid and cells at the site of inflammation. 4. Swelling. 5. Reduction in temperature at the site of inflammation. 6. Pain at the site of inflammation. 7. An influx of leukocytes and phagocytic cells into the tissue. The last option, the migration of immune cells away from the site of inflammation. So the correct option here would be, we have to select which of these are not physiological event during inflammation, and the correct option would be option 5, that is reduction in temperature at the site of inflammation. And last option, that is migration of immune cells away from the site of inflammation. So inflammation typically causes an increase in local temperature that leads to heat due to vasodilation and increased blood flow, not a reduction. And the migration of immune cells away from the site of inflammation, the last option, immune cells migrate towards the site of inflammation, not away from it. All the other listed options, that is vasodilation, increased capillary permeability, influx of protein-rich fluid causing swelling, pain at the site, and leukocyte influx, are all classical physiological events in case of an inflammation. Now next question, Which of the following is not an action of the complement system? You have to select two correct options here. Options are trigger inflammation. Option 2, repel infiltrating leukocytes from the site of infection or inflammation. Option 3, attract phagocytes by moonlighting as chemoattractants. Option 4, attract phagocytes by moonlighting as chemoattractants. Option 5, promote the attachment of antigens to phagocytes. Option 6, protect foreign objects or organisms from degradation. Option 7, cause lysis of gram-negative bacteria in human cells displaying foreign epitopes. Last option, remove harmful immune complexes from the body. So the correct options here would be repel infiltrating leukocytes from the site of infection or inflammation and protect foreign objects or organisms from degradation. These two options are not correct actions of the complement systems. The complement system usually attracts leukocytes to the site of infection via chemoattractant fragments like C5a. It does not repel them. Next, the complement system functions to promote the clearance and destruction of foreign pathogens, not protect them. It opsonizes pathogens, promotes phagocytosis, and forms membrane attack complexes to lyse the bacteria. The complement system's action include triggering inflammation, attracting phagocytes, promoting antigen attachment to phagocytes, opsonization, causing lysis of gram-negative bacteria, and removing harmful immune complexes from the body. Here is the next question. Which of the following systems is directly involved in the initial response to biomaterials when they come into contact with blood? Options are adaptive immune system, endocrine system, complement system, or respiratory system. The correct answer here would be the complement system. The system that directly is involved in the initial response of biomaterials when they come in contact with blood is the complement system. When the biomaterials contact the blood, the complement system is rapidly activated through the classical, alternative, and lectin mediated pathways, and it leads to processes such as protein adsorption, generation of inflammatory mediators C3a, C5a, recruitment of immune cells, and promotion of coagulation. The complement system acts as a key mediator in the immediate immune and inflammatory reactions to biomaterials, unlike the adaptive immune system, endocrine system, or respiratory system, which are not directly implicated in this early phase. So option C, complement system, would be the correct answer. Here is the next question. Which of the following is not a function of inflammation? Options are delivery of leukocytes, concentration of toxins, initiate repair processes, or vasodilation. Vasodilation. The correct answer here would be option B, concentration of toxins. It is not a function of inflammation. Inflammation functions to deliver leukocytes to the site of injury or infection, to initiate repair processes, and involves vasodilation to increase blood flow and immune cell access. However, inflammation does not concentrate toxins. Rather, it aims to remove, neutralize, or isolate harmful agents, including toxins. Moving on to the next question, which of the following best describes the role of foreign body giant cells in a foreign body reaction? Options are: they are responsible for acute inflammation and rapid tissue repair. Option 2, they result from fusion of macrophages and are involved in long-term reactions around implants. Option 3, they form fibrous capsules around implants within hours of implantation. Option 4, they are primary component of granulation tissue and initiate the complement cascade. The correct answer here would be option 2. They result from fusion of macrophages and are involved in long-term reactions around implants. So the foreign body giant cells form by the fusion of macrophages in response to large foreign material such as implants. They are key players in the chronic phase of the foreign body reaction and remain at the biomaterial surface for prolonged periods. They attempt to degrade or isolate the foreign material and contribute to chronic inflammatory response, which is distinct from acute inflammation and rapid tissue repair. And they do not form fibrous capsules within hours, nor do they primarily initiate the complement cascade to form granulation tissue. Moving on to the next question, which pathway of the complement system is most relevant for biomaterial interactions? Options are: classical pathway, lectin mediated pathway, alternate pathway, or the mitochondrial pathway. Correct answer here would be alternative pathway. It is amongst the pathways for the complement system. This is the most relevant for biomaterial interactions. It is primarily activated due to the spontaneous hydrolysis of C3 and its covalent binding to the surface. This activation is amplified by the formation of C3b on the surface, which can bind factors B and D to form the C3 convertase, leading to further complement activation. While the classical pathway can also be involved, especially if antibodies or immune complexes are present, the alternative pathway is generally considered the main pathway responsible for complement activation on biomaterial surfaces because of its spontaneous activation mechanism and lack of requirement for prior antibody binding. So the most relevant would be alternate pathway. Next question, what is the main concern with fibrous capsule formation around drug delivery implants? Options are it promotes rapid drug diffusion into the body, it leads to an increased rate of drug release, it prevents immune cells from accessing the implant, it creates a diffusion barrier limiting drug release, and therapeutic efficacy. The correct option here would be it creates a diffusion barrier limiting drug release and therapeutic efficacy. So the fibrous capsules are formed as a part of the foreign body reaction and consist of densely packed collagen and fibroblasts, which reduce the effective release rate and bioavailability of the drug. This capsule can negatively impact the sustained and controlled delivery of therapeutics, potentially leading to reduced efficacy of the implant. Therefore, it prevents drug diffusion and release, not promotes rapid diffusion or increased release rate, nor does it primarily prevent the immune cells from accessing the implant. So that was all the questions. Thank you for this session.